Welcome back to the deep dive. You might be winding down your week, but in the labs and the regulatory bodies across the world, the pace of discovery just never seems to slow down.
It really doesn't. We've been
Okay.
looking at the last, 48 hours of pretty dense medical journals, your NEJMs, your Nature Medicines, and a few things really jumped out.
Yeah, and we've tried to synthesize the key movements, the things that are really reshaping how we approach human health.
You've got three stories today, and what's so fascinating is just their sheer breadth. They really map the entire spectrum of modern medicine.
How so?
We're starting with a super refined, personalized cancer therapy. Then we're jumping to this bold venture into genetic level regeneration, and we land on a massive, almost accidental public health surprise.
So our mission today is pretty straightforward. We want to pull out the three most critical, maybe most surprising nuggets of knowledge that just hit the wire. This deep dive is really your shortcut to getting up to speed on where medicine is headed. To give you a clear map, we're focusing on, first, what we're calling a new milestone in conquering blood cancer. It's a T-cell therapy getting another big approval.
Then we'll get into RNA that reverses heart damage, which is,
Yeah.
well, it's a glimpse into true organ regeneration.
And finally, my favorite, a vaccine that accidentally prevents dementia. This is the kind of unexpected finding that could change millions of lives.
It really could.
Okay, let's unpack this. And let's start with the most immediate clinical win, this monumental approval from the FDA.
Yeah, what's fascinating here is how quickly some of these personalized therapies are becoming, standard care. We're talking about the FDA approval from December 4th.
Okay.
For Bristol-Myers Squibb's RRT therapy, Breyanzi. The generic is lisocabtagene maraleucel, but we'll stick with Breyanzi.
And this is a big deal because it's fourth indication, right? It's cementing its place.
Exactly. It's becoming a fundamental tool for fighting certain blood cancers.
And we should be really clear about who this helps, like right now. This is specifically for adult patients with relapsed or refractory marginal zone lymphoma, or MZL. Now, for the learner in our audience, MZL is a slow-growing cancer, but it gets incredibly serious when it stops responding to the normal stuff.
The standard chemotherapy or CD20 antibodies, yeah. For those patients who failed those therapies, the clinical reality was often just a devastating prognosis. Very, very few options left.
So that's why these trial results are so important. It's not just a small improvement.
Oh, it's a whole new lease on life. The data comes from the Transcend MZL trial, and you tell them the numbers.
Okay, let's talk numbers because they're really compelling. The overall response rate, the ORR for this really difficult to treat group, was a staggering 80%.
80%. And even more importantly, the complete response rate, so CR, where the tumors just completely vanish, you can't detect them, that was about 60%.
60% in a group that's exhausted almost every other option. That just shows you the power of this approach.
It's profound.
And it wasn't just the depth of the response, it was the speed too. How fast were patients seeing this benefit?
The median time to response was just 1 month, which is incredibly fast for these kinds of advanced cancers.
Wow.
And it's durable. Over 70% of the people who got that complete response were still disease-free a year later. The lead investigator actually said this offers a potential curative opportunity.
That's a huge shift in language,
It is.
moving from palliative to curative. So let's demystify the mechanism a little. How do you take a patient's own cells and basically turn them into these tumor-seeking missiles?
So the process is called autologous CRT. It's actually, it starts pretty simply. You collect the patient's own T cells from their blood
Okay, so a blood draw, basically. Then they get shipped off.
to a specialized facility. And in that lab, the T cells are genetically modified. They're given this new tool, a chimeric antigen receptor, or CIRR.
And that's the personalized part.
Exactly. That CIR is the new navigation system. It's engineered to find and lock onto the CD19 antigen, which is all over the surface of these lymphoma cells.
Then what?
Then they multiply those modified cells, those special forces, and infuse them back into the patient. They immediately start hunting and destroying any cell with that CD19 marker.
Now, the sources mentioned something unique about Breyanzi, this strict 1.1 ratio of CD4 plus and CD8 plus T cells. Why is that ratio so important?
It's highly deliberate. Think of it this way. The CD8 plus cells are the killers. They do the immediate destruction.
Right.
But the CD4 plus cells are the helpers. They help the killers persist. They create long-term memory. So by controlling that 1.1/1 balance, the idea is you get a powerful initial punch and a more sustained, predictable response with fewer side effects.
That balance sounds critical, but we have to talk about the reality of this. It comes with boxed warnings. What do you need to know about the safety?
It's powerful medicine, and it needs intense management. The two big risks are cytokine release syndrome, or CRS, and neurologic toxicities.
And CRS is basically the immune system going into overdrive.
Essentially, yes. It's a massive inflammatory storm. Now, Breyanzi is considered one of the safer options. Severe CRS is under 5%. But the risk means this can only be done in certified expert centers where they can monitor you 24/7 for days.
So a curative opportunity, but a highly specialized one for now. Okay, that's a great overview of precision immunology. But let's use that idea, turning the body into its own repair shop, as a bridge to our next story. If CAR-T is about teaching the body to fight,
Our next breakthrough is about teaching the body to rebuild.
and this is where we move from the clinic to some really fundamental science, a breakthrough from Cedars-Sinai published just yesterday. And it challenges that core idea that heart tissue, once it's damaged, is just gone for good.
We've all been taught that, right? The heart doesn't regenerate. After a heart attack, you get stiff, useless scar tissue, and that leads to heart failure.
But this new work introduces a novel RNA drug called TY1 that seems to be able to kickstart repair right at the genetic level.
That is the big shift. Instead of trying to inject new cells, which is where a lot of research is focused, this approach tries to awaken the cell's own repair machinery.
And in their animal models, mice that had heart attacks, the results were, well, they were pretty extraordinary.
Extraordinary is the right word. They saw a functional improvement of over 40% in the heart's pumping ability, the ejection fraction.
That's not a small change. That's moving an animal from severe heart failure back towards recovery.
And they verified it on a molecular level. They saw a 65% reduction in these markers for DNA double-strand breaks inside the heart muscle cells.
And those breaks are what lead to the cell death and the scarring.
Exactly. Fewer breaks means more surviving working tissue.
Yeah.
And in the study, that correlated directly to much higher long-term survival rates for the treated mice.
So how does TY1 do this? If the cells don't normally regenerate, what is this RNA telling them to do?
So the researchers figured out that healthy heart stem cells produce a very specific kind of non-coding RNA. It doesn't make a protein or anything. It's a pure instruction manual.
And TY1 is basically a synthetic copy of that manual.
Precisely. It's a mimic. When you get it to the damaged heart muscle, it activates the cell's ultimate repair crew, the DNA damage response system. It supercharges the pathways that reconnect broken DNA strands.
So by patching up the DNA really fast, the cells don't get the signal to die off.
Right. You prevent that programmed cell death, apoptosis, and you reverse the whole cycle of scarring.
The lead investigator pointed out that this could apply way beyond the heart if accumulated DNA damage drives other organ failures.
You're seeing the big picture. What if this isn't unique to the heart? What if this is a kind of universal cellular instruction for regeneration?
It's a colossal implication. But we need to balance the excitement with reality. What are the big roadblocks here?
Delivery. It's always delivery.
All right.
This is all preclinical. It's in mice. Getting that RNA, probably in an nanoparticle, safely to the damaged heart tissue in a human before the liver breaks it down, it's a massive engineering problem we haven't solved yet.
So human trials are still a ways off.
At least 2 to 3 years is the estimate.
Okay, plenty of time to watch that space. Now let's pivot. From the hyper-specific and theoretical to a massive public health finding that came out of just enormous pools of data.
This is the ultimate aha moment in public health research.
Our third story. Can a very familiar vaccine, one for shingles, actually be moonlighting as a protector for the brain?
The question is, can it prevent Alzheimer's? And a huge observational study in Nature Medicine suggests a really compelling link.
And to give this credibility, you have to look at the scale. They analyzed anonymized medical records from over 200,000 people, all over 50 in both the US and the UK.
A huge data set. And they compared 3 groups. People who got the newer shingles vaccine, Shingrix, people who got the older one, Zostavax, and the people who were unvaccinated.
And the key finding.
Those who received Shingrix had a statistically significant 17% to 25% lower risk of being diagnosed with Alzheimer's or a related dementia over a 6-year follow-up.
That is an eye-opening number. And it was particularly interesting that the protective effect seemed to be stronger in women.
It was. Which, it raises the big question. Why? Why would a shot to prevent a painful skin rash seem to be protecting the brain? There are 2 main hypotheses floating around.
Okay, let's start with the most direct one, the viral reactivation hypothesis.
Right. Shingles comes from the same virus as chickenpox, a herpes virus, VZV. It lies dormant in your nerves for decades.
And as we get older, our immunity drops and it can reactivate.
Exactly. Sometimes without even causing a rash. And this low-level chronic reactivation is thought to trigger long-term neuroinflammation in the brain. And that inflammation is a key driver of the plaques and tangles you see in Alzheimer's. So the vaccine suppresses the virus, and you remove the inflammatory trigger.
That makes sense. It ties the protection directly to what the vaccine's supposed to do. What's the other theory?
It's called the trained immunity hypothesis. Shingrix uses a very powerful adjuvant booster called AS-ILDO1B to create a huge immune reaction.
Right.
The theory is that this big, systemic immune stimulus might non-specifically train the brain's own immune cells, the microglia.
So the whole immune system gets a tune-up, and that has this off-target benefit in the brain.
That's the idea. This training might make the microglia better and more aggressive at cleaning up the garbage, like the beta amyloid plaques, slowing down the disease process.
The potential here is just staggering. One of the neuroepidemiology professors quoted our sources said that if this is confirmed in proper clinical trials, it could be the single most cost-effective anti-dementia intervention in public health history.
It's incredible to think about, but we have to apply some critical thinking here. This is an observational study,
Meaning correlation, not causation.
not proof of causation now.
And what's the biggest statistical trap they have to worry about?
It's the healthy user effect. It's a classic problem. People who are proactive enough to get a shingles vaccine also tend to be more health conscious in general.
They exercise more, eat better, see their doctor.
All things that could also lower their dementia risk, totally independent of the vaccine. Now, the researchers try to control for those factors statistically, but it's really, really hard to separate the true vaccine effect from that healthy behavior bias.
So we have compelling evidence that screams for more research, but we need to wait for the ongoing randomized controlled trials, the RCTs, for a definitive answer.
Absolutely, that's the next step. But even as a correlation, it shows you the kind of potential that might be hiding in plain sight in our existing public health tools.
So let's just synthesize these three breakthroughs. We started with SART, a super precise, personalized cancer treatment that's already here.
Activating the immune system to fight.
Then we went to the bold venture of TY1 RNA for the heart, which is trying to activate the cell's own internal repair crew.
Right.
And we finished with this massive surprise with the Shingrix vaccine, which might be accidentally activating a protective immune response against dementia.
The common thread, really, is activating our own internal potential. We are moving away from just treating symptoms to actually instructing the body's own machinery T cells, DNA repair systems, microglia, to heal itself.
And if we go back to that TY1 heart repair story for a second, the RNA that patches up DNA breaks, that principle seems so fundamental.
It is. I mean, if that approach works by revving up the DNA damage response in one organ that doesn't normally regenerate, what does that imply for other organs that fail because of that same accumulated wear and tear?
Like chronic liver disease or kidney failure?
Exactly. Where scarring and cellular aging from DNA damage is the endgame. So the question you should be mulling over is this. Which other major organ systems might be next in line for this kind of awakening treatment, one based on fixing our own DNA from the inside out?
A stunning prospect for the next decade of medicine. Whether it's the personalized cell cure, the promise of genetic self-repair, or the surprise hiding in a standard vaccine, we encourage you to keep exploring these fascinating areas. We'll see you next time for the next deep dive.